All right. Well, thank you so much for inviting me. It's wonderful to be here. Just real quick, can you hear me in the back with the microphone okay? Is this volume okay? Okay, excellent. I strained my voice yesterday giving a talk, so if it's a little bit quiet, feel free to call me out. Um, all right, let's see. So, come up. Okay, great. Um, so I'm not totally sure why I got invited to this because my physics background is probably worse than the chemistry background that you mentioned. I think you lost me at Hamiltonian after that. My eyes rolled back. Um, so we're going to talk about some exciting small molecules, though, since it sounds like everybody here is very passionate about new drugs. So before we get started, I thought I'd give a quick introduction to what Drug Hunter is and why we're covering these sorts of topics. As Drug Hunter is drug discovery distilled. The problem that our company is trying to solve is every day companies are disclosing new strategies, tactics, setbacks, unusual toxicities, new targets. But all of this knowledge is fragmented across an increasingly dilute base of thousands of journals, conferences, patents, databases, SEC filings, FDA documents, and so on and so forth. Making it impossible for human scientists to find this information and use it. What we do is we have a team of former industry scientists that go through all this information and make it more digestible for people with a range of different backgrounds supporting small molecule drug discovery. Today, we support over 200 organizations worldwide, including most of the top pharma companies and most biotechs. And again, what makes our team really unique is this is a service that's really created by industry scientists for industry scientists. Pretty much all of us have worked in both biotech and pharma companies from Pfizer to Lilly to Amgen and Novartis, and bring that sort of expertise to the coverage that we provide. One of the things that the team did that I'm most proud of actually is we were commissioned by Bio, an industry organization, to put together a summary of why small molecules are so valuable and different from the drugs of the past, so that Bio could take this to Congress and explain why pills don't deserve 9-year, short life in this IRA negotiation versus biologics. There's sort of this misperception that small molecules are simple, that they're easier to make than biologics, things like that. So some of this draws from the work that we did there. So in the current environment, I'm sure everybody's feeling this a little bit with all the negative news that's been going on in the industry over the last couple of years. So much so that I've started to hear questions like, are small molecules dying? Is this a modality in decline? So I thought I'd start this talk with a quick recap and remind folks that this is a very common phenomenon in this industry. This is a industry that goes through a lot of mood swings. Every 5 or 10 years, the sort of flavor of the month changes. ADCs were sort of no man's land 5 years ago after the Stemcentrx failure. Now, thanks to Daiichi Sankyo, all of a sudden, they're the hottest thing ever again. But there's one thing that's undeniable. It's that drug discovery has made a tremendous amount of progress. So this slide shows a selection of fully synthetic drugs from 1970. And so after the moon landing, we had drugs like mitotane and ketamine. And it's remarkable to see that, aside from natural products, which were primarily either semi-synthetic or grown from fermentation, fully synthetic drugs were quite limited in structure and size. Fast forward 20 years, by the 1990s, right? You can see that fully synthetic drugs had evolved a little bit, got a little bit more complex. You had more than one ring in molecules, but still relatively simple compared to what the molecules today look like. Now, heading into the 90s, there was a lot of consolidation in the pharmaceutical industry. The industry was moving away from natural products-based drug discovery. There was a lot of concern that maybe there wouldn't be big opportunities anymore now that antibiotics were no longer a big deal. We sort of had bacteria, treated bacteria like a solved problem. But of course, there were still blockbusters that emerged in different indications, things like cardiovascular disease instead of antimicrobials, right? Mega blockbusters like Lipitor started to come about with over $10 billion a year in sales, worldwide sales in 2007. Now, around this time, shortly after, there's this great article that came out in Forbes in 2011 by Matt Herper. I think he's now a prominent journalist at Stat News. And people probably remember in this room probably remember the feeling in 2008 to 2011 when a lot of headlines were coming out like this. titled "The Blockbuster Drug Comes to an End." Full stop. Right? No qualifiers. Just it's all over. And this is almost 15 years ago. And one of the sentences I really like from this is at the end it says, in November, Lipitor, the last branded drug among the 15 most used medicines in the US, goes off patent, marking the end of the blockbuster era. So did blockbusters disappear? Obviously, I think we all know the answer, absolutely not. When we look at the top selling drugs just last year, we saw Keytruda, PD-1 inhibitor, a checkpoint inhibitor for immuno-oncology, did a whopping almost $30 billion a year in sales, nearly triple that of Lipitor. We had Apixaban, number 4, Eliquis, that's a typo, that's for clotting, not for oncology, also at over $10 billion a year. HIV combo, helping contribute to the control of the what was formerly an HIV epidemic, at $11 billion a year. And this is something fascinating as well. So as I've been giving talks at many different companies around the world, I've been asking people, how many people remember when HIV was considered a lethal disease? Right? You know, show of hands. I think often I've been surprised to see that it's less than half now. And in some companies I've gone, you know, maybe a quarter of people raise their hands because younger generations of people actually are not taught or don't even remember that HIV was this scary, terrifying disease. And now thanks to drugs that not only control the disease, but prevent it from spreading. You know, it's truly remarkable what the industry has done here. And from a sales perspective, there were 37 blockbusters in 2007. There were over 60 small molecule blockbusters in 2023. And so that's adjusting for inflation. So I use the cutoff of drugs that did over $1.4 billion a year in sales 2 years ago. The number of blockbusters has more than doubled. The size of blockbusters has grown. So by all measures, blockbusters have not gone away. They've grown bigger. We might be witnessing the birth of the mother of all blockbusters, the GLP-1R agonist class. This is a nice picture of Lilly stock price over the last couple decades. I always like to point to this graph because you know, some of the folks in this room may remember, less than 10 years ago, investors were deriding Lilly as this boring company that was still focusing on areas that nobody cared about, like metabolic disease and CNS drug discovery. Well, it turns out now the two hottest areas in drug discovery are metabolic disease and CNS drug discovery. The top 5 GLP-1 products are on track to do over $50 billion a year in combined worldwide sales in a single year. For context, Novo at one point was valued at almost 400 billion. The GDP of all of Denmark in 2024 was about 400 billion. So it's kind of interesting context there. Okay, so blockbusters have not gone away, and hopefully with some of these specific examples, we'll show that the future of small molecule drug discovery really has never been brighter. I don't think small molecules are dying. They're not going away. I think they're going to come back with a vengeance. So one of my favorite areas to start with is GPCR drug discovery, because everybody learns that GPCRs are, easily druggable with most drug class, and yada yada. That must mean it's a solved problem. But actually, there's a lot of exciting stuff going on in GPCR drug discovery. So when people think about, easily drugged GPCRs, they're often thinking about class A GPCRs that tend to have endogenous small molecule ligands like adenosine, binding pockets that are more accessible. But actually, some of the most valuable drug targets have been class B GPCRs, which typically have these big extracellular domains. They bind to long peptides that are very difficult to simulate with small molecules, and have at some points been classified as impossible to drug. So only in the last couple of years have we started to see drugs approved here. So the CGRP antagonist class, famous, 2-decade-plus story to get to antagonists that could be orally given for migraine. Parathyroid hormone receptor is another class that remains technically undrugged, although there are clinical candidates that have emerged. And now we're finally starting to see small molecule GLP-1R agonists take the news. So you may have seen this molecule in the news recently, or orforglipron. I think it sent Lilly stock up again, as if it couldn't, go any higher, and sent Novo down because it is the first oral GLP-1R agonist to show compelling data in phase 3 for diabetes, and it showed weight loss as well. So implies that it will likely be useful for obesity. In this molecule, I always joke that if you're not a chemist, you can get a sense of how hard a target was to drug by looking at how gnarly the structure is. This thing is, to a chemist, quite beastly, almost 900 molecular weight. Yet it's an oral molecule, despite having 15 hydrogen bond acceptors, 8 to 11 rings, depending on how you count, and nearly 900 molecular weight. It's an oral molecule with 9 to 45 mg once daily oral dosing. And surprisingly, even though it has some reactive groups like an indole, an amide, a THP ring, it has a 30 to 50 hour human half-life. So this small molecule is exciting and has some of the peptide developers a little bit worried because it could not only be more efficacious, more convenient for patients versus injection, lower cost, more scalable, combinable, and co-formulatable, especially as new combination options come on, versus oral peptides. One of the things that people don't talk about with these oral peptides as well is bioavailability of semaglutide, for example, in the Rybelsus formulation is around 1%, which means you need 100 units of drug to get 1% into the bloodstream. And the cost, when you're talking about an indication where 1 billion people a year potentially could take this kind of drug, there's not enough starting material on the planet to manufacture that amount of peptide. Not only that, these injectables, even though they're not as inconvenient as these injections used to be, the injector pens actually are a supply limiting reagent, I guess you could call it at the moment, where these pens are expensive to manufacture, inspect, and so on. So small molecules don't have any of those problems. This could be a very exciting area. And of course, Novo now, I've heard, is investing heavily in small molecule discovery, even though it's for a long time been a peptide company. I've heard that they have a goal, actually, of having at least a third of their portfolio come from small molecules within the next 10 years. Now, so I visited Chugai a month and a half ago, and I asked them, how come you guys haven't disclosed this? And they apparently have a draconian publication policy, so you never see them at conferences, you never see their papers, and every so often they drop a bomb like this in the literature, and you're like, wow, where did that come from? We did ask them, how was it to discover the drug? And the head of discovery there said one word, said painful. So I'm like, okay, I believe you. So we can get a sense of how hard this was from Pfizer's campaign. So Pfizer, of course, has as good a screening platform and as great scientists as anybody has in the industry. If you read some recent reviews, it's been said that they've had interest in GLP-1 since the 70s, had small molecule programs as early as the late 90s, and we're tried everything to find chemical matter for this receptor. They ultimately had to rely on a really creative approach to find starting points, and I think this is a nice thought experiment for how computational approaches might help in the future. Instead of screening against the native receptor, what they did was they pre-activated it by treating it with a covalent positive allosteric modulator, BETP. This forms a covalent complex that is more sensitive to small molecule activators, and presumably because some kind of conformational change. And then they took this and screened with this. And you can imagine this is a very risky strategy, as if you screen with this covalently modified species, you might only get hits that work with that covalent activator. And then if you remove that covalent activator, it's a totally useless drug, right? Fortunately, they were able to find reversible hits that were greater than 10 micromolar in the presence of this BETP reagent and were not active as expected. Without that covalent modulator. Fortunately, with some elegant medicinal chemistry, they were able to optimize these compounds to get them down to single-digit nanomolar activities, both in the presence and absence of this covalent positive allosteric modulator, leading to clinical candidates like danuglipron. Now, what I like about this is when you look at this compound on the right, people talk a lot about chemical space and sampling chemical space. I think sometimes there's been a little bit of false perception that because we've made all these high throughput screening libraries with millions of compounds, or now we have DEL with billions of compounds, that we've somehow mined out drug-like chemical space, and we have to go into beyond of rule of 5 chemical space. I think this illustrates that that's absolutely not true. The universe of even drug-like chemical matter, the chemical space that exists even below 500 molecular weight, is so unfathomably massive that we've barely scratched the surface there. And a lot of times it's how do you set up the assays? How do you interrogate your target that can be limiting? So beautiful example of a traditional rule of 5 compound for a previously considered undruggable target. Now, thanks to cryo-EM, it seems like we're able to see complex structures of molecules bound to transmembrane receptors more frequently. I think 10 years ago, we'd be lucky to see a cryo-EM structure in Nature once a quarter. Now it seems like every time a new molecule gets disclosed, it's a cryo-EM structure is deposited in the PDB right away. To give you a sense of how quickly the industry is adopting this, when we were in Japan, we heard several companies already had cryo-EM facilities internally. I heard BMS here is trying to build out their own cryo-EM facility, and Deerfield, surprisingly, in New York City, is installing a cryo-EM machine in the basement of one of their Manhattan buildings. So that was very surprising to me. But it's being quickly adopted, and hopefully this will lead to more structural information that'll be available to folks focused on free energy approaches and things like that. And now we can finally see how molecules actually bind to receptors like GPCRs. It was a very black box 20 years ago. This molecule, surprisingly, only appears to form two discrete interactions with the receptor, a hydrogen bond with the ether ring at the top left over there, and an ion pairing interaction with this carboxylic acid isosphere at the bottom left. And everything else kind of seems to fill space and fill lipophilic space, which is very surprising. Another dimension that cryo-EM is helping with now is for these previously black box targets like GPCRs, finally starting to get some structural insight into how different molecules behave, right? So what challenge with GPCRs was always, if you're playing with antagonists or agonists, sometimes you make small modifications, they suddenly switch to antagonists, which is not what you want. With agonists, you can have partial agonists, full agonists, biased agonists, et cetera. And again, in the past, it's very trial and error. You make a molecule, see what it does, and if you're lucky, it does what you want. Now with comparative, comparisons between I keep accidentally saying crystal structures, comparisons between cryo-EM structures, we can now get an understanding of, well, why is a molecule a partial agonist? Why is it a full agonist? Danuglipron, the Pfizer molecule, is a full agonist, and the hypothesis is that's because it induces a conformational change in this helix TM7, whereas orforglipron, which binds to the same binding site, surprisingly, even though they're two completely different molecules, orforglipron is a partial agonist and doesn't activate the receptor to the same extent. Who knows which one's going to be better in the clinic? So far, orforglipron is performing better, not for efficacy reasons, but for tolerability reasons. But I think this level of resolution could be impactful for understanding all kinds of different receptors. Obviously, the opioid receptor has been a target that people have been exploring for a long time and trying to see, can you come up with biased receptors that only have pain relief but no addiction and no constipation? I'm sure there are other receptors as well where this kind of structural understanding will help come up with new program ideas. As far as structural enablement goes, there's sort of a catch-22 here, which is now that there's more structural information, it's getting a lot easier for companies to come up with follow-on programs and make molecules that behave similarly but are structurally different and have their separate IP. So a great example of this is aleniglipron from a Chinese biotech, Structure Therapeutics. Structure Therapeutics stock went up to, I think they were valued at almost $4 billion at one point because of this molecule. People viewed them as a credible competitor to the Lilly molecule. And you can see here that really all they did was they swapped out this left-hand group for an interesting diethyl phosphine oxide. I'm sure if you model this, it probably behaves very similarly to orforglipron. But this is now a very credible competitor that apparently has its own freedom to operate. What's interesting about this as well is you can imagine that with this increased structural information, this is not only going to be true of hot targets like this, but of everything else in the GPCR universe where maybe people thought they had an advantage by not disclosing assay data or how things work, but now the structural information might help people understand that faster. This is funny. So we talked to the medicinal chemists at Structure Therapeutics. Another trend recently has been the emergence of Chinese biotechs. And we asked them, well, gosh, these molecules are quite complex. You guys must have hundreds of chemists in Shanghai or whatever. And they're like, oh, no, we only have 10. It's mostly us and a couple modelers. I was like, you guys don't have labs? And they're like, no, we don't. I'm like, well, how do you get anything done? I said, oh, we outsource everything to Wuxi. So I guess it's not too hard to copy that. All right. Another interesting area is mRNA display screening coming of age. And this is another exciting area for, I think, modeling and free energy approaches to make an impact as well. So everybody's probably seen this resurgence in interest in peptides. Why is that? Well, there are a couple examples recently of oral clinical candidates emerging with macrocyclic peptide cores, either fully synthetic or based on traditional peptide backbones. What is mRNA display? mRNA display is a screening technology that's very similar to DNA-encoded libraries thematically. So similar to DNA-encoded libraries, how you barcode members of the library with an oligonucleotide. You can generate billion-mer libraries of macrocyclic peptides using this approach. But the challenge here was medicinal chemists thought that this was a technology that could only be used for generating in vitro hits that bound to the target, but would not be useful for making cell-active compounds and certainly not for making orally bioavailable drugs. Why is that? When you look at the structures of the hits that come out of this, not surprisingly, because they're macrocyclic peptides, they're big, they're hairy, they've got lots of polar residues, very easily cleaved, not things that look easy to optimize to be oral drugs at all. So in the last couple of years, when Merck reported oral PCSK9 peptides, Protagonist reported oral IL-23 peptides, BMS reported oral PD-1 peptides, and several other companies reported interesting oral compounds like for RAS from Chugai or others. The industry sentiment kind of turned and say, okay, well, maybe generating oral clinical candidates with this technology is not just feasible, but is something that we should invest more heavily in. This molecule is 1,550 molecular weight. This is Merck's PCSK9 macrocyclic inhibitor that they reported had oral efficacy in phase 2 of 2023. 30 mg once daily of this molecule leads to significant reductions in cholesterol in patients, and it is in phase 3 for hypercholesterolemia. What's remarkable about this fundamentally is when you think about a small molecule, this is quite massive compared to your typical small molecule. However, when you think about antibodies, which are typically 140 kilodaltons or greater, this is quite tiny, right? This is 1% the weight of an antibody, yet it has potency comparable to the clinical PCSK9 antibodies, 5 picomolar binder versus 8 picomolar for the antibody. So now we're getting into territory where fully synthetic molecules are able to directly compete with antibodies on binding sites that traditionally were thought to be only accessible to antibodies. So people are talking about, small molecules versus biologics. I'd be worried if I were the biologics because now there are all these oral synthetic alternatives that allow you to do very similar things. This is also fundamentally interesting because apparently this molecule is cell impermeable and yet it's orally active. And so how is that? Well, a lot of companies working with these peptides use permeation enhancing lipid formulations like sodium caprate. Prevailing hypothesis is this may help with paracellular transport through the gut. Because this molecule is so potent and has such a long residence time against this extracellular target, it doesn't need to be in the bloodstream in very high concentrations to have its oral activity. So this is the first orally active clinical candidate from this mRNA display technology. You have to give the Merck and Raw Pharma teams a ton of credit. Raw Pharma was a biotech here in the Boston area that had done some pioneering work on this platform. They got hits like this molecule on the left, and they didn't give up. They said, we're going to keep going with that. And I think most people, I don't know what your sense is of chemistry, but for most medicinal chemists, if, in our first job, we got that molecule put on our desk and we said, okay, here's your project for the next 5 to 10 years. Your job is to make that orally bioavailable. Good luck. I think most medicinal chemists would be applying to other companies. Maybe it's more work for modelers, and that's a good thing, but I don't know. So for mRNA display, for a starting point of 1,800 molecular weight and 1 micromolar is definitely a brutal challenge to make this more orally available. They were able to get to more potent compounds like lead compound 50, Ki of 24 nanomolar. And at this point, they really wanted to get co-crystal structures to confirm that these things were actually binding to PCSK9. Before this, they didn't have co-crystal structure. It's just showing activity in this assay. But as you know, there are all kinds of ways you can show activity in assays without binding to your target. Fortunately, they were able to get a co-crystal structure. And this is super cool. When you look at how the lead binds to the target, One thing that you'll quickly see is that there's no binding pocket, right? Uh, Often when we think about how small molecules bind to targets like enzymes, you imagine that these molecules fit inside some kind of groove, or there's some sort of volume that encapsulates these molecules. The protein doesn't fold over this molecule at all. It's just kind of floating on top of it like a pancake, right? This is very similar to a protein-protein interaction. When we think about how we evaluate drugs targets for druggability, right? One of the parameters that we're looking for is there a binding pocket? Is there a volume? Is there lipophilic space that can be filled by a small molecule? I think examples like this are suggesting, well, maybe there's a whole lot more ways that we can drug all kinds of biomolecules that don't necessarily depend on traditional pocket being there. So how do we define binding sites? That may change in the future. The Merck team with Raw Pharma was able to pull off a really cool triple macrocyclization approach to get these molecules down from double-digit nanomolar binders to single-digit picomolar compounds. What you'll notice with the triple macrocycle when it's overlaid with the starting point is that the triple macrocycle doesn't form any new enthalpic interactions with the target at all, right? That triazole that's introduced as a bridge is solvent exposed. The alkoxy residue on the left is solvent exposed. And so this 10,000-fold increase in potency is purely driven by conformational restriction and reduction of entropy of binding. So again, when we're thinking about free energy approaches and how this can be powerful, if you can get 10,000-fold increase in potency just by making sure that your conformation is minimized, not just enthalpically, but also entropically, that can be an extremely powerful approach to increasing potency. All right. So this all sets the stage for why AstraZeneca's disclosure of another Boston area molecule acquired from Dogma Therapeutics, AZD0780, was a big deal. This is a traditional looking small molecule that also displayed remarkable clinical data on targeting PCSK9. So they saw 52% reduction in LDL cholesterol when taken orally versus placebo in phase 1. In combination with statins, this led to almost 80% reduction from untreated in baseline versus 66% maximum for the Merck molecule. And so now we have a head-to-head competition between a traditional small molecule, which may be cheaper, lower cost of goods, and more accessible, versus a macrocyclic peptide that Merck believes is going to be more efficacious. And maybe both will be used in different ways. We'll see. Okay, another exciting example for the mRNA display technology is LUNA18. This is an oral pan-RAS inhibitor from Chugai in phase 1 for cancer. In contrast to the Merck molecule, this one actually is traditionally cell penetrant. So it is able to get into cells and target an intracellular target RAS, which is also considered undruggable for a long time. And this, without using any special formulations, has 21 to 47% oral bioavailability across species. So this is the first clinical candidate from mRNA display where scaffold hopping from the original core was not required. And this was the goal of mRNA display technology all along, was can you get hits, optimize them with medicinal chemistry, not have to do any crazy core changes or anything like that, but just optimize the macrocycle hit that you get and turn it into an oral clinical candidate. And that's exactly what Chugai did here. And that's what's one of the reasons that everyone in the industry is excited, because if Chugai can do that for this impossible target, where else can we apply these approaches? All right, another area that is seen sort of a resurgence is covalent drugs. And there's a really cool molecular glue I'll talk about in this story as well. So covalent drugs have gotten a bad rap for some time for potential safety liabilities. Now, it seems that the perception of these molecules in industry has changed over time, as in the last couple of years, we've seen several multi-billion-dollar acquisitions for covalent molecules. And importantly, these acquisitions are not taking place in oncology, which typically has a lower, perceived lower bar for safety than other disease areas. But these are taking place outside of oncology for indications like sickle cell anemia or chronic diseases like multiple sclerosis. Mavaloxolone is a cool molecule because this is a reversible covalent molecule with a highly reactive cyanoenone warhead. Most people assume that these sorts of things would have a short half-life because of their reactivity. They would get quickly chewed up by GSH. Turns out this has a 57-hour average human half-life. And one of the reasons for that is it's a reversible covalent inhibitor. GSH does not form stable bonds to this molecule. It's reversible. It only forms a stable conjugate with its intended target, keep1, thanks to the interactions with the actual receptor. Voxelotor is a super cool example of an aldehyde-containing drug. Aldehydes are pretty rarely used as covalent warheads. They're perceived to be highly reactive and not very safe functional groups to use. Although when you think about it, glucose and all of the sugars in our blood are actually masked aldehydes, right? So we actually have quite a high concentration of aldehydes floating around at all times. But this drug is super interesting because Pfizer is one of the most conservative development organizations out there, or had a reputation for being very conservative, having a very high bar for safety. And they paid $5 billion for this drug to develop in sickle cell anemia. This drug not only has an aldehyde warhead targeting an N-terminal amine, which is quite interesting, not a lysine. It is dosed 1.5 grams once daily, very high dose, is intended to be used chronically, targets one of the most abundant proteins in blood, hemoglobin, has a 10-to-1 blood to plasma distribution because it gets soaked up by the hemoglobin, and is intended to be used chronically in an indication where it's predominantly in adolescents, or predominant in young adolescents. So for Pfizer to take an aldehyde at high dose chronically in kids, presumably forever, it's a pretty radical development move. So I think it's representative of how the times have changed with respect to this modality. Another one of my favorite examples illustrating how times have changed is Pfizer's ritlecitinib, a JAK3 covalent kinase inhibitor. Again, for Pfizer to take a covalent kinase inhibitor outside of oncology into alopecia areata, a hair loss condition, is a sign of the times, because hair loss is certainly not as severe as oncology indications. One of my favorite stories from the recent covalent drug discovery space comes from Boston area company, Relay Therapeutics. I think there are a couple of people here from Relay, right? Anybody here in the audience? I saw some people wandering around. Anyway, this is one of my favorite stories. Super cool program. So the FGFR family of receptors have been implicated in a wide range of different cancers, and everybody's screened against FGFR, but finding selective inhibitors of FGFR1, 2, 3, and 4 has been very challenging over time. FGFR2 in particular, people had not found reversible ATP competitive molecules that were highly selective for 2 over 1 or 4. And the challenge is that each of these isoforms carries on-target toxicities. So if you inhibit FGFR1, you see hyperphosphatemia. If you inhibit FGFR4, you see severe diarrhea. So when Relay said that they had the first isoform selective inhibitor of FGFR2, a lot of chemists paid attention. They said, wow, Relay must have found like a cryptic pocket or an allosteric molecule or something, because everybody's screened against the ATP binding pocket and nobody's found selective binders. So when they revealed the structure at AACR, a lot of chemists looked at that and said, okay, there's no way this thing is selective. It's got a hinge binder, a couple arrow groups, looks very planar, has a acrylamide. There's no way this thing is selective across the kinome. But you look at their data, and you can see that the molecule is almost 300-fold selective in cell-based assays for FGFR2 over 1. And the proof is in the pudding. When you look at the clinical data, they have a much greater objective response rate in FGFR2-driven cancers versus the approved pan-FGFR inhibitors. Yet, unlike the pan inhibitors, they see no significant clinical signs of hyperphosphatemia due to FGFR1 inhibition or severe diarrhea due to FGFR4 inhibition. So how on earth is this molecule functionally selective? Well, their hypothesis is that this is due to molecular dynamics. So if you look at the binding sites of FGFR1 or FGFR2, the residues involved in binding the molecule are basically identical. So the binding sites are virtually 100% conserved. There's almost no difference in how these two receptors bind the molecule. So where do you get this dramatic selectivity from? Well, they say that when they did molecular dynamic simulations, they saw that the N-terminal lobe, the northern part of this protein, oscillates much more quickly or dynamically in FGFR1 whereas in FGFR2, it's less dynamic and oscillates more slowly. And so there's a cysteine on that northern part of this protein that they alkylate with this molecule. And so the thinking there is that that cysteine is more accessible to alkylation in the less dynamic protein than in the more dynamic protein. So the binding site is exactly the same, but the dynamics of the protein are different. For chemists, another way to think about this could be, it's a reaction, right? It's an alkylation reaction of the cysteine. The entropy of alkylation is higher for the more dynamic protein, lower for the less dynamic protein. That's a energetic way to think about this, perhaps. When you look at their kinetic data, you can see that the Ki, a measure of the reversible affinity of this molecule for the binding site, is only about threefold different. It's a weak binder to both isoforms, 6 micromolar for FGFR1, 2 micromolar for FGFR2. But the rate of inactivation, the K inact, is almost 30-fold greater for FGFR2 over 1. And so combined, this leads to a 90-fold greater efficiency of inactivation of 2 over 1, leading to the functional selectivity in spite of the near total conservation of the binding sites. So super interesting example. And they say that this helped discover this molecule in only 18 months from starting the program. All right, looks like, are we doing on time? We've got about 9 minutes, 5 minutes? Okay, all right, so you can't talk about covalence without talking about KRAS. So you all know that there are 2 approved KRAS G12C inhibitors now, and Revolution Medicines has a new platform that is really exciting. They have molecules that target KRAS in a completely different fashion. So the way these molecules like RMC-6291 work is rather than binding KRAS directly, the way these work is they first bind to an intracellular cytosolic protein, cyclophilin A. That binary complex is then able to form neoprotein-protein interactions with the intended target KRAS, leading to a tri-complex and inhibition of KRAS. So this is a super cool mechanism, and this has roots in Boston as well. So folks who have read The $1 Billion Molecule and know about the history of Vertex may know that Vertex wasn't founded on cystic fibrosis or ion channel work. They weren't originally interested in HCAB. Actually, they got started working on natural products like FK506 and rapamycin, which are natural product molecular glues. They didn't get anywhere with the structure-based design of these molecules, but what everybody in the industry had hoped at some point was that chemists would be able to synthetically re-engineer these natural products to act as molecular glues, not for their native substrates like FKBP12 and calcineurin, but for intended substrates like KRAS, as in this Revolution example. The Revolution chemical matter derives from a natural product called sangliferin. This was discovered in a Novartis antimicrobial library. Turns out to be a molecular glue for cyclophilin and IMP-DH2. A local company called Warp Drive Bio set up a covalent fragment screen or a covalent tethering approach to look for molecules that bound to both KRAS as well as cyclophilin A, and were ultimately able to come up with leads that look a bit more drug-like, like this molecule on the right. To cut a long story short and a ton of work short, it's really impressive that Revolution Medicines, the team there, was able to optimize these compounds to bind to both substrates, KRAS as well as cyclophilin A. And you can imagine that with this type of drug discovery, where you're dealing with not just one protein, but 2 protein targets, free energy approaches might be really powerful in helping make these intricate design decisions where you're not only worried about one interaction, but 2 at the same time. As if targeting KRAS G12C was not enough, Revolution has come out with several exciting new candidates, all targeting previously undruggable targets like KRAS G12D. As recently as 2022, people were saying the chemical leverage of aspartic acid by covalent KRAS G12D inhibitors will likely be impossible, which I'm sure the Revolution med chemists laughed at because they probably had this molecule in-house at that time when that was published. But this is a covalent G12D targeting molecule that uses an aziridine warhead to covalently alkylate a carboxylic acid residue. It is orally bioavailable in humans with once daily dosing, despite its massive size. and impressively has shown a 30% overall response rate in phase 1 with a 1.2 gram once daily dose. There have been more recent disclosures from Revolution Medicines. Now they say they even have a reversible, non-covalent inhibitor of all forms of RAS, not just KRAS G12D, including KRAS G12V, G13X, Q61X mutants. And this has been really inspiring because they have not only seen response rates in terms of stable disease and partial responses in phase 1 in pancreatic cancer. They've even seen a complete response in a 77-year-old patient with stage 4 pancreatic cancer. This is the type of thing that was unheard of or literally thought to be impossible 20 years ago. Now, thanks to newer chemistry, newer modalities, and some really bold drug discovery by various companies, we're starting to see the impossible take place in a lot of ways. Okay, so since we've got 5 minutes left, I think we'll leave that there. I'm sure everybody's excited about targeted protein degradation and all the complex pharmacology there as well. It's just going to create a lot more work, I think, for free energy folks. So I think we'll just leave that there for now. Okay, so if I leave you with anything today, I hope it's that despite the mood in the industry, despite everything that you're hearing about, small molecules, maybe going out of fashion or funding and all this stuff, there really has never been a more exciting time for synthetic molecules in drug discovery. There's just so many other stories I didn't have time to talk about today, but it's just incredibly exciting. So with that, thank you for the invitation. I know we're running short on time, so. Absolutely. I think there are a lot of companies that do that. I think C4 Therapeutics or C4X Therapeutics and other companies, their entire company is based on studying macrocycles. To clarify the point about the reduction in entropy, it's I'm not saying that the binding of the molecule is primarily due to reduction of entropy. It's more the increase in potency relative to the starting point is due to the reduction of the entropy binding between those two molecules. But absolutely, I think this would be super helpful. I think some of this stuff, when I think about it as a medicinal chemist, how we think about these things, a lot of it is, well, we think this might restrain it. We don't necessarily calculate it, but we're going to take a wild guess. And then, 80% of the time it doesn't work and 20% of the time it works. I think if you could involve the calculations earlier, if it were more accessible or easier, it would certainly be valuable to have an estimate of, well, is this actually going to enforce a conformation we want? Is that conformation actually going to be bioactive? Those things would certainly help save a lot of synthesis because these macrocycles are rarely easy to make. Great question. I think if I were to boil it down to a single factor, it would be minimum efficacious dose, right? So minimum efficacious dose kind of, aggregates everything that you need, the PK, the expected target coverage, all that stuff. I think that's, what makes drug discovery hard is, there's 100 variables that go into contributing to what is the minimum efficacious dose. But if you were able to, summarize that all in one parameter, that would be kind of it. Love it. That comes up a lot. So I think it's a really fun discussion to have. When you think about where did the rules even come from, and why did we even have rules in the first place? A lot of this history comes from the wave of combinatorial chemistry, right? So after natural products, and that sort of inspiration, people started to make these big combinatorial libraries, and the medicinal chemists would get these libraries from the CombiChem folks, and they'd throw them like 10,000 molecules, and they'd all be ugly, and the chemist said, okay, stop that. We don't want to, and maybe that's similar to how virtual screening is taking place now, but that's an aside. They'd say, okay, we have to give you some criteria for the kinds of things you're making through this CombiChem approach, because not all of these CombiChem things are worth making, and they're not all worth certainly screening. And so I think a lot of these rules, rule of 5, rule of 3, all these things were really meant to be guidelines for the library chemists. And then they happen to be useful, later on for guessing, okay, what's more likely to be orally bioavailable, less, et cetera. But if you talk to Chris Lipinski or any of those folks, these were never meant to be like, they were meant to be guidelines. They were never meant to be like rigid, don't do this. That said, I think the principles are still useful, right? Like statistically, if you make a compound that's 1,000 molecular weight, statistically, it's probably still going to be less likely to be bioavailable than something that's 200 molecular weight, but doesn't mean it's impossible. And I think where the industry has shifted in the last 10 years is the doesn't mean it's impossible qualifier has just become a bit stronger, and people are more open-minded to checking more things. That's a great question. So sorry, you were talking about the program with D. Shaw, or? Oh yeah, I think they've published 2 papers on this in the AACR journals. Yeah, I couldn't tell you off the top of my head. Sorry. Yeah, I'm sure. Yeah. Is there anybody from D. Shaw here? Maybe they can comment as well. Yeah, so I think that is actually the real surprising part of this. So, these big molecules, like these big macrocycles or, bifunctional molecules, people have been talking about these for ages and ages. What was really surprising in the last 10, 15 years was the human proof of concept showing that you can actually have oral molecules in man with these sorts of things. And I think the DMPK folks, there's been a lot of discussion on how do you predict this. And I think it boils down to, well, a lot of the rules still apply. If you have something that's more quickly metabolized or, cleared by transporters or whatever, permeability, all these things, they all still matter. It's just that, if you're more willing to check in this space, you might be surprised to find some things are more permeable than expected due to things like chameleonicity or multiple conformations. Or other cases where there's some other things that are unexpected going on that lead to overall properties that still allow you to have oral human availability. That said, back to the rules thing, right? Many of these are still exceptions rather than like, I think if you talk to the folks working in degraders, it's still hard to come up with oral bifunctional molecules. It's just not impossible.